we are going to discuss about the cell wall. So it is the outermost boundary of the living cells like plant cell, algal cell, bacterial cell and cells of the fungi. And accordingly they show different compositions. It is absent in case of animal cell. What I am saying, it is present only in the plant cell, algal cells, fungal cells and bacterial cells. So here these cells, they show different compositions of the cell wall, which is the outermost boundary, that is outer to the cell membrane. This is present. So this cell wall in case of algae or in case of plants, it shows the materials like cellulose, even hemicellulose is there. Many times it is associated with proteins and lipids in addition to the pectin, that is pectate. Calcium or magnesium pectate is present in it. In case of fungus, it is made up of special substance, we call it as chitin. In case of fungal cell wall, chitin is the material. While in case of bacterial cells, we have studied while studying the prokaryotic cell, the bacterial cells, they show the cell wall having peptidoglycans. That is the material in case of bacterial cell wall. So we will see the common structural arrangement of the cell wall that is formed over here. So here the shape or rigidity, the firm shape of the cell is because of these cell walls, where we observe the three layers in case of plant cell wall. So starting from the outermost layer, we'll see what are the actual layers. So the outermost layer, we call it as middle lamella, which is the common layer between two neighboring cells. Here we are observing one cell. In the neighboring another cell is there. So it is the common structure present between the neighboring cells. Here another cell will be there. Here another cell will be there. So it is the common layer between neighboring cells. We call it as middle lamella. Below that, the second layer is there. We call it as primary wall, primary cell wall, which is the first wall produced, and it is increasing in surface area. It is thin layer and we call it as primary wall, that is second layer, inner to the middle lamella. Below that, the innermost layer is present, which is thick layer, and that we call it as secondary wall layer, secondary cell wall layer. So these three layers are there, and in between the secondary wall layers we observe some microfibrils, where deposition of the cell wall material is very less or it is thin, that we call it as plasmodesmata, where microfibrils are present. Same structure you can observe vertically. It was the top view of the cell. If you observe it in side view vertically, you can get an idea that here neighboring cells are there. So cytoplasms of neighboring cells plasma membranes of neighboring cells, and in between these two the layer called middle lamella is there, followed by the primary wall layer and secondary wall layers. So traversing of the plasmodesmata, which is the connectivity between neighboring cells, we observe them as in the form of microfibrils. We call them as plasmodesmata. So these three layers, we'll see their composition in details. So the middle lamella, what we call it as common layer between neighboring cells, it shows composition, that is, it is formed by calcium and magnesium pectate. It is formed by pectate of calcium and magnesium. Next to that, the primary wall layer, that is the first wall which is secreted by the protoplast. जी प्रोटोप्लास्टनं सेल डिव्हिजन झाल्याबरोबर जी वॉल सिक्रेट केलेली असेल, that is first wall. It shows cellulose, non-cellulose, that is non-cellulosic polysaccharides are present. Even hemicellulose is present. Also it shows lignification and it increases in surface area. Whatever size of the cell, that surface area is due to this. Below that, the secondary wall layer, which is more in thickness, it shows almost same composition, that is, it is having same, that is cellulose, non-cellulosic polysaccharides, hemicellulose, and at maturity it undergoes lignification. It is present in the cells like sclerenchyma or even in the trachees, the trachea of xylem, and it may show deposition of suberin. So it undergoes more thickness during maturity. The functions of the cell wall, whenever we observe this boundary, it is definitely having some functions, that is the protection to the inner contents of the cell, that is by the cell wall material. Even it is giving rigidity to the cell where they are present. Rigidness of the cell is due to the cell wall only. Even it is useful for the transport across neighboring cells. We have shown the presence of plasmodesmata. In between, these connections are there which are maintaining the cytoplasmic connectivity between the neighboring cells. So all these functions, they are carried out by the cell wall, which is very important structure.
Now,
after the discussion about cell wall, we are switching to the biomembranes, or what we call it as plasma membrane or cell membrane. So it is the common membrane which is present surrounding the cell, as well as the membranes of the cellular organelles. They are also of the same kind. So collectively we call them as biomembranes. There is membrane of the cell as well as the membrane of the cellular organelles is of the same kind. So this biomembrane, it was described by various scientists, and from all those views, the most accepted view in the entire universe, that is universally accepted model which describes all the characters of the biomembrane, that is fluid mosaic model. It was suggested, it was described by the scientists Singer and Nicholson in 1972. So according to this model, that is fluid mosaic model, the nature of the membrane. It is quasi-fluid. That is, you can compare it with the icebergs in the ocean, where two layers of lipids which are arranged here. That is, by layer of the lipids, they are arranged with heads on the outer side and their tails are facing inner side. So here the globular or spherical heads of these lipid molecules, which are polar in nature and they are hydrophilic. They are having affinity for water, while the non-polar tails, which are facing towards inner side, these are hydrophobic in nature. They are not having any affinity for the water. They are hydrophobic in nature. So in between these two layers of the lipid, the protein molecules are embedded irregularly. That is why it is called as mosaic. Or right now what I said, that is icebergs in the ocean. So it is fluid in which the protein molecules are embedded, which are irregular in there, and they may change their position. Their positions are not fixed. They may change their positions. So here the arrangement, some proteins, they are totally inside in these two layers. That embedded entirely in these two layers of the lipid. That is, starting from the lower layer, they are going to pass through the entire length. So overall the thickness of this membrane is 75 angstrom units. So these are passing this entire length. They are the integral proteins and they may be working as the channels from one end of the membrane to the another end. So the proteins which are totally inside, we call them as integral proteins, while some proteins, they are working as the membrane surface proteins. The surface of the membrane, it contains some proteins which are on the outer side. These are called as peripheral proteins. So altogether it is working as the membrane or the boundary surrounding the protoplasm, or what we call it as cytoplasm, and collectively we are calling it as biomembrane. So this ratio of lipids to proteins, it varies in various cells. For example, in the RBCs, the percentage of proteins we observe is about 52%, while the lipids are 40% in this composition. So here overall arrangement is like where the non-polar tails, they are never in the contact of the aqueous fluids which are present near these membranes. So these membranes, these are very important part of the cells as well as the cell organelles. They are performing various functions. So the first function of this membrane is that it allows the entry as well as substances. That is, some substances it allows inside as well as some substances it allows the exit. So that is the function where the substances, they may be passively going or coming in. That is, against with the concentration gradient they are flowing, we call it as simple diffusion. So that is the first function. Sometimes the substances which are crossing the membrane against the concentration gradient, they require energy in the form of ATP molecules, and that we call it as active transport over here. The non-polar regions, they are not allowing the movement of the polar molecules across the membrane. The another function of the membrane is that it can recognize its own type. It is even contributing to the immune response, that is, it is working as the barrier in our immune system, as well as it is involved in the acceptance or rejection of the transplanted organs, that it can recognize its own type. Even what I said, it is involved with the help of the surface proteins or peripheral proteins which are working as the membrane proteins. For example, the mitochondrial membranes, they show presence of the enzyme proteins which are involved in the electron transport chain. Those are membrane proteins or the peripheral proteins. So with all these important functions, it plays very significant role in the cellular activities. Even it is involved in the carrying or transport of the messages in the form of entry of some biochemical substances. That is the all about the biomembrane or the cell membrane which we observe in case of all living organisms.